All right, what is regulation, Matthew? Control. So in part 2 of the cell cycle notes, we're all the way down toward the end. Regulation or control of the cell cycle. Control for normal growth and development. Regulation varies for each type of cell. Because depending on what type of cell you are, then you may have to divide every day or two, or you may not ever divide, in the case of most neurons and muscle cells. The regulation is controlled by proteins called cyclins. So cyclins are the proteins that are going to fluctuate and going to tell the cell basically when to divide and when not to divide. 3 checkpoints exist during the cell cycle. If you look on page 243 at the top, you have a checkpoint toward the end of G1, making sure that the cell is big enough and it's mature, has all the organelles and things that it needs. There's a checkpoint at the end of G2 to make sure that you have all the components needed, so the cell should have been growing bigger, it should have been making more organelles, it should have DNA copied, all of those things before it ventures into M phase. And then during metaphase, you've got another checkpoint to make sure all the chromosomes are there, they're all lined up in the middle, they're all ready to divide and ready to go. Okay? All those checkpoints are important in regulating the cell cycle. Why do you think the cell would what would happen if the cell didn't have checkpoints in place? Nate?
There'd be a lot more mistakes. Growth process.
Yeah, it'd be a lot more mistakes. A lot of cells that divided that weren't supposed to, cells that divided didn't have all their chromosomes lined up and everything ready, so you would have a lot of mistakes in daughter cells if that was not there in place to make sure everything went all right. All right, part V. What's the difference between theta chromosomes and eukaryotic chromosomes? Theta means what? All you Greek people out there? It's a circle, good. So that circular chromosomes, like in, why is the circular chromosome? Prokaryotes. And eukaryotes, which have linear chromosomes and multiple ones. Part A, Walker.
More genes are present on eukaryotic chromosomes than on theta chromosomes.
Keep going.
Keep going. Okay. This means there exists more genetic variation exists in eukaryotic organisms. There also exist more possible genetic combinations that can be inherited.
All right, so eukaryotes have a lot more DNA, a lot more possible variation and variety. Obviously, we're going to have more genes because we're more complicated than prokaryotes. Now, I will give this to prokaryotes. A prokaryotic chromosome is almost all of it is coding for something. So it's very efficient in its DNA. Whereas our DNA, we may only have 2 to 4% of our DNA that actually codes for specific proteins or things that are made and produced in our body. We've got a ton of pseudo-DNA, a ton of old viral DNA and stuff that no longer codes for anything. Part B, Matthew.
There exists more genetic stability in eukaryotic chromosomes. Offspring receive the same number of chromosomes most of the time. Important linked genes tend to be inherited together.
Yeah, we'll talk about linked genes and chromosomal numbers a little more when we get to genetics. Part C allows for diploid cells to exist, the result of sexual reproduction, and one half of the DNA to come from each parent. Sexual reproduction is significant because it helps us to create much more genetic diversity. The more diversity you have, the better off you are, the more likely you are to survive and do well in diverse environments. Any questions about part 2?
I have kind of a random question.
Yeah.
So you're talking about how muscle cells don't really divide, and like that dude who had like the chunk of his calf taken out that never really repaired. Like, do muscle cells not have the ability to divide and like kind of repair like that, or?
Not that I'm aware of. Once you're born, I don't know. I don't think so. I don't think you can make them go back into that state. Muscle cells are kind of weird though. Some are multinucleate, meaning they have like multiple nuclei. Because some of them can get like muscle fibers can get really big, really long. So they're unique. And then we've got 3 different types of muscles. We have Cardiac, we have smooth muscle, which is all of our stuff that's in a circle, like arteries and veins in your stomach and stuff. And then we have, skeletal muscle. I'm not aware of a situation where they would start dividing again. If you find some, you can look it up for extra credit and tell us about it, but not that I'm aware of. Now, I mean, like in the future, maybe they'll have develop stem cells from people and then implant that, and that we could develop that into more muscle tissue.
That's what I was wondering if, like, maybe in the future they could figure out a way to, like, make those reproduce in a way that could heal.
Yeah.
Things like that.
Some people have figured out how to take skin cells and turn them back into pluripotent cells that can become different types of cells. Maybe not all different types, but some different types of cells. So I imagine that's something that they'll continue to work on. The problem is, I would think it's very expensive, and there's a lot of people, so I don't know if they get to the point where it's so efficient, it's something everybody can have access to. All right, part 3, important concepts. Enzymes are proteins that catalyze and regulate cellular processes. Catalyze means that you help a process take place. Regulate means to control. Enzymes, almost all enzymes are proteins. There are a few enzymes that are made out of RNA. We call those ribozymes. But most all are going to be proteins doing the work and helping the reactions take place throughout your body. You guys turn to page 244. These two figures on 244 are pretty important. I can imagine y'all seeing these again on a test or quiz. Something very similar to them because they illustrate what's going on in the cell cycle, okay, and how it is regulated. Cyclin production. Remember, cyclin is crucial to when the cell goes into division, okay? This protein concentration amount increases from S phase until anaphase. So if you look at the top of 244, cyclin is the purple line from the middle of S phase all the way through the middle of M phase. That is a steep increase in cyclin production, okay? So what's going on from S phase toward M phase? M phase. Well, I know, but what's fixing to happen? It's getting ready for the cell to divide, yeah. Or the cell is then dividing into M phase. Cyclin must combine with cyclin-dependent kinase to become active. So, you're going to have cyclin, which is a protein in the cell. Its concentration is increasing as it gets toward division. It has to combine with cyclin-dependent kinase, which is a type of what? Enzyme. It's going to help something happen. So those two have to combine here. Yes? All right, read 1 and 2 for Sarah.
Kinases are enzymes that turn on processes within the cells. Remember, enzymes are not used up, so concentration of kinase remains constant.
All right. This isn't going to be the only time you hear the term kinases. These are enzymes that are turning on active processes throughout a cell, a lot of times like a cellular cascade where one process turns on another. You'll hear these a lot when we get into the next section on cell signaling. So these are constantly being reused. The only thing that's really varying here is the amount of cyclin production throughout the cell cycle. All right, part 3. Ashlyn, get your life together. 5. Go, quick.
Together they make maturation promoting factor MPF. This is a process the cell wants on. MPF causes the cell to undergo mitosis when levels are high within a cell. Concentration levels must reach a check point, levels begin. Cyclin starts being produced in the S phase and keeps being produced until anaphase begins.
Okay, so a couple things here. One, let's make a math equation out of this. What is the math equation you should be able to get from that section of notes?
Cyclin plus CDK equals MPF.
Good. Cyclin plus CDK equals MPF. Now, if you look at the bottom picture here on 244, you'll notice that cyclin's accumulation increases from S through the middle of M phase there, and that cyclin then starts to combine with CDK. When the two combine, they make what's called MPF, maturation promoting factor. It's promoting the maturity or development, or in this case, the division of this cell. Okay? And let's say cyclin is the main contributor there. Number 4, Tay, read that for us.
After subsequent phosphorylation of cyclohexylation and nuclear blood players to start breaking down for conversion into spinal apparatus.
All right, phosphorylation, what does that mean?
Add phosphates to ATP.
What? We didn't say it's ATP, it's just adding a phosphate. It could be the ATP, but not in this case probably. Phosphorylation, you're adding a phosphate for the purpose of what? Energy. Provides energy. Phosphates are little energy packets that you throw on different molecules. Phosphorylation of cytoskeleton and nuclear envelope occurs to start breakdown for conversion. So the phosphorylation will be energy for the breakdown of the nuclear envelope and also converting it into the spindle apparatus, which we use to help the nucleus separate. Cyclin degrades after mitosis, leaving only CDK behind. The enzymes are recycled or reused. So after division takes place, you can see that cyclin production just completely drops off. It's broken down until the next go around that it's needed when more is produced. Okay? Number 2, kinetochore signaling. This signal occurs at the 3rd checkpoint at the end of metaphase. So after all those chromosomes have lined up in the middle, they're ready to divide, to split apart. What happens? Read B for us, Mason.
As chromosomes, kinetochores
Kinetochores.
connect with the spindle apparatus, enzymes are turned on.
All right, so around each chromosome you have a centromere, and kind of the outside part of that where it connects with the spindle apparatus is called the kinetochore. Those enzymes are turned on. Now, you're going to see another one here. This the enzymes are called anaphase promoting complexes because they're promoting anaphase. I would say that one's not near as important as the maturation promoting factor, cyclin, and cyclin dependent kinase. And to be honest, the name's pretty self-explanatory there. When concentration levels of APC reach a checkpoint, anaphase begins. Chromosomes have attached to spindle fiber. They're ready to begin anaphase. Promote means to start. Complexes means several molecules working together. So they start the separation of sister chromatids during anaphase. Okay. Part 3, density-dependent inhibition. Inhibition means what?
Like stop that separation.
Slow or stop it. Density-dependent means what?
It's dependent on the density.
It is dependent on the density. Turn over to page 46 in your 246, sorry, in your books. Mariah, read A and B for us. Yeah, under density-dependent inhibition.
Basically means a cell stops dividing when contact with other cells is made. Seen in a pair, once no one is sealed, cells back in contact, they stop dividing to fix the wound.
Okay, so what happens if you get a cut somewhere on your body?
Blood.
What does that do to the skin? Scrapes it. What else does it do? It splits it, right? Take it away. I hope you didn't get that big a scar or that big a chunk taken out of you. So if you get a cut somewhere on your skin, it cuts the skin in two. And the skin's response, the cells are to do what?
To like fix, like patch up the hole.
To patch up the hole, to fix the gap. Okay, so the density-dependent inhibition means when there's a gap or a break, your cells are going to start dividing because they're not in contact with one another all the way around. And they're going to keep dividing until they come in contact with one another again. And they form what we would call a scar, where new tissue has formed there. If you look at your scars, I got a pretty big one on my elbow here from a fencing accident. If you look close at it, you don't see all the same like hair, and you wouldn't see probably the same glands and stuff. It's mostly just your basic skin tissue. but that is done to repair those wounds and keep that area from getting infected and hurt even worse. Okay, so that's density dependent. Now you can see the same kind of thing at the top of 246 here. They take a few cells, they put them in a petri dish with a nutrient solution. Those cells will continue to divide until they come in contact with other cells. Another example of density dependent inhibition. Anchorage dependence. Read that one for us, Katie.
Basically means cells must be connected to connective tissue base to divide.
Wound.
Wound severity is key. If a hunk of flesh is cut off, it won't be connected or anchored and be able to get nutrients soaked out.
Okay. Anchorage dependence. We're not talking about Alaska here. This means your skin or cells need to be anchored to something else. And really what it's talking about is you have to have underlying blood vessels and connective tissue to support the growth of new tissue. So if you got in a bad car accident or you got cut really bad or a wild pack of dogs got a hold of you or something like that, and they ripped a big enough chunk of skin away from you and underlying connective tissue like the dermis and hypodermis, then you wouldn't be able to grow new skin cells on top of that because there wouldn't be the nutritive support in place there. And a lot of times you may have to have some kind of surgery. You may have to have stitches or staples or something like that, so that there is time and energy needed to basically somebody else is helping you put that tissue back together. Okay. All right. Cancer, abnormal cell growth. Onco means cancer. If you go to DCH and you go to the parking lot up second or third floor, what do you see? A lot of spots reserved for oncology. Oncology is study of cancer. Good. So the oncology unit's up usually, I think, at the second or third floor. And so if you see that, that's study of cancer. Now, it's not probably a good idea when people, or it's not really accurate when people say, like, I want to cure cancer, or let's find a cure for cancer. That's not really an adequate saying. Why?
Because cancer
Is
is
this
like isn't necessarily a disease. It's more of like a mutation or abnormality.
Well, what else? I mean, you can't just cure cancer. Like, you can't come up with a magic pill and cure cancer. Why?
Just cell division.
Well, I was thinking the main reason is there's a lot of different causes of cancer. So you can't, it's not like HIV where, like, you get rid of one virus and you're done. There's different things that cause cancer. Throw it over there, Gage. Thanks. Appreciate it. So it's not as simple as that. Now, there's roughly 80% of all cancers that are caused by two mutations to two main genes, the P53 and RAS genes, which we'll get into, I think, more when we get to genetics. But just saying, like, let's cure cancer, it's probably not plausible just because of the different causes of cancer. I think the number is somewhere around for women, 1 in 3 will get cancer in their lifetime, and guys, it's about 1 in, I want to say 1 in 2 maybe. They say about 1 in 6 men will get prostate cancer, the most prevalent one, although probably one of the more treatable types of cancer. So these are prevalent. I'm sure you probably all know someone in your family or around you who's had cancer. And young people can get cancer. A lot of kids have leukemia. A lot of guys get testicular cancer in their teens and 20s. I had a student who had cancer a long time ago. A student actually died probably 5 years or so ago after she left school, or had to, I think it was right after she graduated. So cancer can affect people of all different ages and backgrounds. All right. Cancer creates abnormally high cyclin production within the cell. No checkpoints exist within cancer cells, so there's no density-dependent inhibition. Cancer cells are considered immortal as long as oxygenated blood is available. Now, if you look on 246, on that bottom right picture, they've shown you what cancer cells would do. So instead of stopping when it comes in contact with other cells, they just keep going and dividing and get bigger and bigger. And we call that a what? Tumor. As Arnold would say, tumor. Got a tumor. So, where was I? Immortal cells. HeLa cells, number 2. HeLa cells prove this, named after Henrietta Lacks. They've been growing since 1951. So in 1951, this lady Henrietta Lacks, they took some cells for her. She had some kind of reproductive part cancer, and it was really aggressive. They took some cells, and I think they were like, hey, we'll grow some in a petri dish and see if we can research these. And they're still growing. You can still get Henrietta Lacks cells today in 2020, even though she died 70 some odd years ago, because her cells are still dividing. They are basically immortal as long as there is enough nutrient solutions available. Here's a book if anybody wants to borrow it, The Immortal Life of Henrietta Lacks. This is more of a book on medical efficacy, about it was really wrong that they took her cells without her consent or her family's consent or whatever. But they have done a lot of good in terms of the research that's been had on the Henrietta or the HeLa cells, as they're called. All right, number 1. You down for Miranda?
Angiogenesis
Mm-hmm.
occurs means creation of new blood vessels to feed the tumor.
Yeah. That's problematic. When you start when you have cells divide to the point where they start undergoing angiogenesis, anytime you see angio, you're going to think blood vessels. You can go get an angiogram, which would be like one of those pictures where they show like the blood flowing through your heart and see if anything's stopped up. Genesis just means to create, or the beginning. So once you have cells that are big enough and there's blood supply to them, then you've got serious problems, because it's not as easy as just going and chopping it out. You've got to make sure that you do something with those blood vessels. Those blood vessels are starving other cells of nutrients. So those are issues when you get to cancer. So the next thing, telomerase, is a type of enzyme. Tell us about it, Nathan.
Where were we at?
Telomerase, under HeLa cells.
Yeah, okay. Telomerase enzyme present. This enzyme is fueling the adenine production of cyclin. Everyone has the ability to make this enzyme because it is needed during development in the womb to make the organism develop quickly. Once the organism is developed, it gets turned off normally. It's turned back on by cancer-causing substances called carcinogens. Cigarette smoke is an example of a carcinogen.
Carcinogen, any cancer-causing agent. So when you're a little baby in the womb, your cells are dividing super duper uber fast. I want to say at some point, like your neurons, they're making like 250,000 new ones a day or something. It's like a ridiculous number how often they're dividing and splitting. But eventually your body has to turn this off. Otherwise, I don't know how big you'd get and how much resources and stuff you'd need to get there. So that's a problem. So if this gets turned back on, then basically that quick division takes place. All it takes is one cell. One cell gets turned on, it starts dividing, and you can have a large mass of cells very quickly, because then it's a cascade. They all divide. They're all got that same DNA or that malfunction, and it's like a domino effect. Excuse me. Cigarette smoke, I want to say there's like 43 some odd carcinogens been identified in cigarettes. Any kind of chemical agent that's been identified that's a cancer-causing substance is identified is called a carcinogen. Yeah.
So how does cancer kill you? Does it starve other cells that need?
Yeah, I mean, so cancer cells are your cells. They've just grown bigger. For instance, like if you got cancer in your brain, eventually those cells would continue to grow and starve out other cells that need to function. Or pancreatic cancer is probably the number one highest death rate cancer. So it gets in your pancreas. Like my father-in-law he was a doctor. It got in his pancreas. It got in his intestines. It got in his liver. It got in his stomach, which is all right together there. And the first thing he knew was he had a little bit of a backache. And I don't know what led him to go get checked out, but when he did, basically it was just too late at that point. He only had a few months to live. So it starts taking over and robbing the other tissues around it of nutrients and kills those tissues that you need to survive. Yeah. So, we continue onward, and hopefully we have a better way of figuring out how to attack and kill and address cancer issues. Now, there's another example. We'll get there in a second. Normal cells divide between 1 and 100 times depending on the cell type. If no telomerase is present, the cell will not be able to continue dividing. I mean, it's really dependent on like what type of cell it is, how often it's going to divide before those cells die out. Cancer starts with transformation of DNA in a cell, transformation of telomerase to the on setting. Now, like I said, there are multiple genes that could cause excessive cyclin production and cause cells to start to divide out of control. It's a lot more complicated than this. When we get to cell signaling, we'll look at a couple of pathways in cells that lead to cancer, a couple of the more common ones. So we'll definitely get more into this. Things that can cause this: a weak genetic history, trauma, viral inserts such as with HPV, and repeated carcinogen exposure. I'm sure y'all been to the beach, see all the people laying out on the beach, getting all the UV rays they can. The more you damage that UV damages your DNA, the more likely a harmful mutation is to occur. Genetic history. I think it was Angelina Jolie had a couple markers for breast cancer, and she wound up getting a double mastectomy, if I'm not mistaken, because of the high likelihood of her developing that. Hopefully in the future, we'll all be able to get tested and be like, you know, you've got genetic markers that say there's a really high chance of you getting like colon cancer. So you would know to go get, you know, a colonoscopy every year or get checked out more often. So to kind of mitigate that problem. Trauma, I guess that maybe, I don't know, chemical, I don't know what trauma they're talking about there. Genetics is a big one, and then environment, probably genetics and environment's probably half and half, roughly. I mean, like, some people could smoke out of their life and never get cancer, and some people could smoke or dip tobacco. You know, you hear baseball players that are 17, 18 years old dip tobacco for a few years and they get mouth cancer and they have to have half their mouth taken off. So it really just depends on your genetic disposition on a lot of that. Let's see here. HPV. HPV is a cancer-causing, we would call it an oncogenic virus. Cancer-causing virus. What do y'all know about HPV? You might get a shot in here for HPV. What do you call the shot? Gardasil? Isn't that what they call it? Isn't that the main one?
I got that when I was like 13.
Yeah, they give it pretty early. So they've developed a vaccine for HPV because it's especially harmful to girls because it can cause what? Nope.
Breast cancer.
Nope. What'd you say?
I said breast cancer.
Nope. When you start going to the lady doctor, what do they test you? What do they give? What do they test? What's the test they do? 3 letters. It's the first 3 letters of the middle word. Pap smear. You heard that? Okay. And so basically what they're doing is they're testing your cervical tissue. They go in and take like a sample, and they test that to see if there's like precancerous or cancerous tissue in there. And so if you have to remove that, then that could cause problems like infertility because your cervix is basically the muscle at the base of your uterus that holds the, you know how they talk about like, you see on TV, they're like you're 5 centimeters dilated or 10 centimeters dilated, that's your cervix. So if that gets cancer and you have to remove that, then I don't know what the likelihood of you still being able to have a child is, but I'm sure it makes it more difficult. And so for a lot of guys, HPV is asymptomatic. Sometimes it causes warts, but for a lot of them, it's asymptomatic. It could be asymptomatic for girls, and you may not know you have it, and you're passing it around. It's probably the most prevalent STD, if I had to guess. Last time I saw the data. All right, so problematic, can cause cancer. Tumor, abnormal growth. There are different types of tumor, benign and malignant. Now, I'm going to wait till tomorrow to go through the last part of this. And actually, we're going to get into those two main types of cancer tomorrow, or what causes, two main causes of cancer. But we'll do that then. Y'all get out your lab packet real quick and turn to that back page.
Does HPV cervical cancer just happen?
You would have to get the virus for that. So if you got HPV, and sometimes, like I said, you may have it, not even know have any other symptoms, that can alter the DNA of your cells.
Wait, certain people are asymptomatic. It's a virus.
So the problem is a lot of people pass it around and never know they had it.
Yeah.
And it's a virus, and so basically what it does is embed in your tissue. It's kind of like herpes, where like it will embed in your tissues, and maybe it gives rise at certain points. It's not cancerous for everyone. Do what?
My mom used to have it, and so
Well, there's a lot of varieties. So then you got to get into like, probably everyone could say they've got like, hold on a second here.